Hi everyone, myself Kesuma studying B.Pharmacy first year in Maharaja College of Pharmacy. Today I want to discuss about a topic that is catabolism of heme from biochemistry. First, synthesis of heme. The synthesis of heme generally involves four steps. Those are catabolic pathway of heme to bilirubin, transport of bilirubin to liver, conjugation of bilirubin and excretion of bilirubin. Next, formation of bilirubin. The RBCs which are present in our body generally have the lifespan of 120 days approximately. After the completion of their lifespan, they enter into macrophage and release hemoglobin. Here, the released hemoglobin releases heme and heme is converted into biliverdin and then to bilirubin. Now we can observe the complete catabolism of heme. The aged erythrocytes, that means RBCs which are present in our body, generally after completion of their lifespan, they enter into macrophage. In macrophage, the aged erythrocytes release hemoglobin. Here, the hemoglobin is divided into two components. Those are heme and globin. Here, globin itself acts as a amino acid and it utilized for the synthesis of hemoglobin. And heme is converted into biliverdin in the presence of enzyme heme oxygenase. And here, the energy is utilized in the form of NADP. Next, the iron is removed in the form of ferric ions and carbon monoxide is released. Next, the biliverdin is converted into bilirubin by the enzyme biliverdin reductase. Here, energy is utilized in the form of NADP. Next, the bilirubin enters into blood and forms a complex with albumin. Here, in blood, bilirubin-albumin complex is formed. From blood, the bilirubin enters into liver and in liver, the bilirubin converts into bilirubin digluconide by the enzyme bilirubin gluconyl transferase. Here, 2-UDP gluconate is utilized and forms 2-UDP. Next, from liver, bilirubin digluconide enters into intestine or kidney in the form of urobilinogen by microbial enzymes. In kidney, the urobilinogen converts into urobilin and which gives the coloration to urine. And in intestine, the urobilinogen converts into stercobilin and which gives the coloration to feces. Next, plasma bilirubin. The plasma bilirubin means the improper levels of bilirubin in plasma is known as plasma bilirubin. The normal plasma levels are total bilirubin is 0.2 to 0.8 mg per dl. The unconjugated bilirubin is 0.2 to 0.6 mg per dl. The conjugated bilirubin is 0 to 0.2 mg per dl. Plasma bilirubin is greater than 1 mg per dl, then it is known as hyperbilirubinemia. If the levels between 1 to 2 mg per dl, then it leads to latent jaundice. If the levels are greater than 2 mg per dl, then it leads to bilirubin diffuses into tissues, producing yellowish discoloration of sclera, conjunctiva, skin, and mucous membrane, resulting in jaundice. Jaundice is generally of three types. They are hemolytic jaundice, hepatocellular jaundice, and obstructive jaundice. Next, conclusion. In recent years, the heme catabolic pathway is considered to play an important regulatory role in cell protection, apoptosis, inflammation, and other physiological and pathological processes. Reference. I refer U. Satyanarayana textbook. Thank you.